Hello dear, welcome to this lecture. In this lecture, we will discuss about ABO blood group system. First of all, we see the contents of this video. In this video, we will see terminologies that are commonly used in ABO blood group system. Then history of blood transfusion. Then blood group system, in which we will discuss about structure of RBCs, RBCs antigens, blood group antibodies, primary and secondary immune response. Then genetics of the ABO blood group system, genetics of different blood group system, synthesis of ABO antigens, how the ABH antigens are synthesized over the surface of RBCs. Then ABO subgroups, then Bombay blood group system that will be discussed in this video. Antisera used in ABO blood grouping. This antisera is prepared, is synthesized by using hybridoma technology. This technology will also be discussed in this video in detail. And then ABO blood groups, how can we do ABO blood grouping? ABO blood grouping is done by different method like slide method, spin cube method, ABO gel grouping. Then ABO blood group discrepancy. All these contents will be discussed one by one in this video. Now we start from first of all terminologies. In terminologies, first one is immunohematology. What is immunohematology? That is very important. The science of immunohematology deals with the basic principles of antigen and antibody structures, genetics, biochemistry, its mode of action, and its role in hematology. That is called immunohematology. Then the next one is immunogen and antigen. Basically, immunogen refers to a molecule that is capable of evoking an immune response by an organism's immune system. But in case of antigen, an antigen refers to a molecule that is capable of binding to the product of that immune response. So an immunogen is necessarily an antigen, but the antigen is not necessarily immunogen. तो इसमें immunogen तो antigen हो सकता है, लेकिन जो antigen है ये immunogen नहीं हो सकता. So that is the difference between immunogen and antigen. Now we explore the antigen. Antigen is a complex molecule whose molecular weight exceed 10,000 Dalton. The ABH antigens, because we are discussing about ABO blood grouping. So in this point of view, the ABH antigens are glycolipids, okay? And RH antigens are proteins in nature, while in case of hepatitis B surface antigens that are made up of lipoprotein. So the antigens may be glycolipids, may be proteins, may be lipoproteins. As for example, ABH antigens are glycolipids, RH antigens are proteins, lipoproteins in case of hepatitis B surface antigen. So the next one is hepton. Hepton actually is a Greek word which means to fasten, to increase. Many low organic molecules, as you can see in this diagram, many low organic molecules having low molecular weight are not antigenic by themselves, but become antigenic if they bond to a larger carrier molecule such as protein. So here in this diagram, you can see the low molecular weight organic molecules that are not antigenic, but the larger carrier proteins that bind with low molecular weight organic molecules as a protein. These low molecular weight antigens are called heptons. So these when these low molecular weight organic molecules bind with the larger carrier proteins, so then it converts into a hepton and also become antigenic Example here is an example of heparin is penicillin, a small molecule that is not antigenic. However, when it is bind with certain serum proteins of sensitive individual, the resulting molecule becomes immunogenic, activates lymphocyte and initiate a severe and sometimes fatal allergic reaction, a type of immune response. So that is the example of heparin. Penicillin. Penicillin is a big example of heparin in which the penicillin by themselves is not an antigenic, but when bind with serum protein, then it converts into antigenic and provokes the immune system and start to make antibodies. So the next one is antibodies. So in antibodies, we will see production, how the antibodies are produced in our body, the structure of immunoglobulins, immunoglobulin classes. There are three types of immunoglobulin that are present in our blood. First of all, antibodies. Antibodies are immunoglobulin in nature. Approximately 82 to 96% of antibodies are polypeptides. ठीक है, जो आपकी हमारी जो एंटीबॉडीज होती हैं वो प्रोटीन इन नेचर्स होती हैं एंड दैट आर मेड अप ऑफ polypeptides up to 82 to 96%, and the rest, the remaining 4 to 18% of antibodies are made up of carbohydrates, or in other words, the 4 to 18% antibodies are carbohydrate in nature. Production of antibodies is done by plasma cell when the antigen or immunogen come into our body. This antigen or immunogen bind with the B cells. B cells then differentiate into plasma cell, and then plasma cells start to make antibodies in our body against the antigen or immunogen. The immunoglobulin structure, when we see the structure of immunoglobulins, all the immunoglobulins share a common chemical structure configuration. Each basic antibody unit is antibody is composed of 4 polypeptide chain. तो इसमें एंटीबॉडीज के अंदर 4 पॉलीपेप्टाइड चेन्स होती हैं, जिस तरह ये आप डायग्राम के अंदर देख सकते हैं, और 2 लाइट चेन्स हैं और 2 हैवी चेन्स हैं. तो ये जो 4 polypeptide chains होती हैं, 2 identical light chains having a molecular weight of approximately 22,500 Dalton, and 2 identical heavy chains with a molecular weight of 50,000 to 75,000 Daltons. And covalent disulfide bonds hold the 4 chains. This covalent disulfide bond is present between the heavy chains and also between the heavy and light chains. So in this way, 4 polypeptide chains are kept together. वो एक दूसरे के साथ उनको वो होल्ड कर लेता है. तो each heavy chain has 440 amino acids. Heavy chains are synthesized due to having 440 amino acids. 440 amino acids are involved in the synthesis of 2 heavy chains, and 220 amino acids that are involved in the synthesis of light chains in immunoglobulins. The chemical structure of heavy chain is responsible for the diversity of immunoglobulin classes. The light chains kappa and lambda are common to all immunoglobulins. So one thing that is very important point, जो हमारी यहाँ पे जो heavy chains होती हैं, ये डिफरेंट होती हैं, और इन्हीं की वजह से इम्यूनोग्लोबुलिन्स के अंदर एक डाइवर्सिटी डेवलप होती है. बट इन केस ऑफ लाइट चेन दैट आर प्रेजेंट आउटसाइड द heavy chain, so these light chains are identical to each other and are not a part of diversity in immunoglobulins. So the diversity in antibody is developed only due to heavy chains. The next one is classes. So classes, immunoglobulin classes. Immunoglobulins are divided. Immunoglobulins have 5 types of immunoglobulins are present in our body. IgA, IgG, IgM, IgE, and IgD. Uh, IgA, IgG, and IgM, these three types of antibodies are a major part of ABO blood groups, major part of ABO blood group system, in which these antibodies are involved. The isotypes of the heavy chains determine the class of immunoglobulins. जो हैवी चेन्स होती हैं, सपोज करें ये IgA है तो इसके अंदर हैवी चेन अल्फा है, IgG के अंदर गैमा है, और इसी तरह जो IgM है इसके अंदर भी ये जो हमारी M जो है इसके अंदर ये हैवी चेन्स हैं, और इसी की वजह से इनके अंदर जो डाइवर्सिटी डेवलप होती है, इसको आगे हम जाके फर्दर भी इनको देखेंगे. There are 5 classes of immunoglobulin designated as IgA, IgD, IgE, IgG, and IgM. The blood group antibodies are commonly IgM, IgG, and or IgA. Or no, the first one is IgA. IgA class, IgA class of antibody exists both as a monomer and as a polymer. The molecular weight is approximately 16,000 160,000 Daltons, and IgG. The IgG constitutes approximately 75% of total serum immunoglobulin. It is a Y-shaped monomer. There are 4 subclasses of IgG: IgG1, IgG2, IgG2, 3, and 4, based on the sequence of amino acids in the heavy chain. So subclasses of IgG actually are based on the sequence of amino acids that are present in the heavy chains. The IgG antibodies react at 37 degrees centigrade, and the molecular weight of IgG is 150,000 Daltons, which is lowest of all immunoglobulins. It emits IgG to cross the placenta barrier. So this antibody is also cross the placenta from mother to fetus. So IgG can react at 37 degrees centigrade. So these antibodies are warm antibodies. The last one is IgM antibodies. IgM antibodies constitutes approximately 10% of the total serum immunoglobulins. So IgG approximately 75%, and IgM is approximately constitute 10% of the total serum immunoglobulins. They are pentamer in shape. IgG is a Y-shaped, and this one is a pentamer, and IgG is a monomer. IgM, they are pentamer in shape. The molecular weight is 9 lakh Dalton, which makes it the heaviest of all classes of immunoglobulin. So IgM is the heaviest antibody present in our body because of having 9 lakh Dalton molecular weight. IgG had a molecular weight of 1 lakh and 50,000, and IgA contained 1 lakh and 60,000 molecular weight. So the IgM is the heaviest antibody as compared to IgG and IgA. IgM, this antibody cannot cross the placenta. These antibodies are not present in a newborn baby, and these antibodies can react at room temperature 20 to 24 degrees centigrade. The IgM are highly effective agglutinants and are capable of activating the complement. This antibody, IgM antibody, is also involved in complement activating the complement, but IgG is not involved in activating the complement system. So this one is also the difference between IgG and IgM. The plasma contains significant amount of IgM. So this is the antibody. We discussed about antibody production, immunoglobulin structure, and immunoglobulin classes. The next one is complete and incomplete antibodies. So what is the difference between complete and incomplete antibodies? The antibodies which are produced without any antigenic stimulus are known as complete antibodies. Complete antibodies mean The antibodies which are produced without any antigenic stimulus are known as complete antibodies. Mostly the IgM class antibodies fall in this category. They are capable of agglutinating red cells suspended in normal saline at 20 to 25 degrees centigrade. Most of the ABH antigens are IgM in nature. Mostly the ABH antibodies are IgM in nature. Keep in mind that the blood group A and blood group B, blood group AB carry about contain about 80% IgM antibodies. But in case of blood group O, the IgG antibody contains 80%. So both antibodies are vice versa in blood group A, B, and AB, as the second one is blood group O. Are IgM in nature and called the natural or complete antibodies. The IgM antibodies are called complete antibodies, and these antibodies are complete antibodies are present in blood group A, B, and AB. No, the incomplete antibodies. The incomplete antibodies, the antibodies that require a bridge like a Combs molecule for binding to the antigenic site are called incomplete antibodies. And the example of incomplete antibody is antibody IgG. So this antibody react at 37 degrees centigrade. The RH, CDE are incomplete or acquired antibodies. So this one is the difference between complete and incomplete antibodies. Complete antibodies are IgM, incomplete antibodies are IgG. IgM antibodies react at 37 degrees centigrade, but the incomplete antibodies react at 20 to 25 degrees centigrade. Complete antibodies are present in blood group A, B, and AB. Incomplete antibodies are present in blood group O. So these are the major difference between complete and incomplete antibodies. The next one is monoclonal and polyclonal antibodies. What is the difference between monoclonal and polyclonal antibodies? Antibodies which are derived from multiple ancestral clones of antibody producing cell and carry both kappa and lambda light chains are termed as polyclonal antibodies. The polyclonal antibodies, those antibodies which are derived from multiple ancestral clones of antibody producing cells and carry both the kappa and lambda light chains are termed as polyclonal antibodies. But in case of monoclonal antibodies have the ability to recognize single antigenic epitope and provide greater diagnostic precision than polyclonal antibodies. Here you can see in this diagram, the right diagram present here, the single antigen attached with the antibody. So this one is the monoclonal antibody, and this antibody is produced from a single ancestral clone, which is responsible for the production of antibody. But here you can see the single antigen having different epitopes. These epitopes are involved to evoke the immune response, immune cells like B cell, plasma cell, and these antibodies are produced from different ancestral clones of B cells, which produced these antibodies and attached with different epitopes on a single antigen. So this one is the difference between monoclonal and polyclonal antibody. But the monoclonal antibody is more preferable for diagnostic, that is used in diagnostic technique in laboratory. The next one is history of blood transfusion. The blood transfusion history, first one is in 1628, Dr. William Harvey discovered the blood circulation throughout the body. Dr. William Harvey, the one of the most old scientists who discovered the blood circulation in our body. After Right after 30 years, James Swammerdam, a Dutch biologist, came and described about the RBCs the first time. And then in 1818, Dr. James Blundell, who successfully transfused blood into humans without any pre-testing, cross-matching, blood typing. In 1901, Dr. Carl Landsteiner, who came and give ABO blood grouping. ABO blood grouping basically was given by Dr. Carl Landsteiner in 1901. In 1907, Dr. Ludwig Hecton, American pathologist, first to describe cross-matching. The cross-matching concept was given by Dr. Ludwig Hecton in 1907 after Dr. Karl Landsteiner, who discovered ABO blood grouping. Then in 1970, right after 10 years, Dr. Oswald Hope Robertson, American pathologist, first anticoagulant citrate glucose was discovered by this scientist. This anticoagulant basically is used to preserve the blood for a long time. As for example, no days, many other anticoagulants have been discovered and more efficient, and we can store by using these anticoagulant for a long time, maximum 35 days. For example, CPDA1 that is used to preserve the blood for 35 degrees centigrade with 80% viability of red cells. Anyhow, in next 1924, Flex Bernstein, American surgeon, who give the inheritance of blood group. This inheritance of ABO blood group system will be discussed in this video later. After the scientist, in 1937, Dr. Bernard Fantus, American physician, first blood bank in Chicago established. Dr. Bernard Fantus, who established the first blood bank in Chicago in 1937. After this, in 1940, Dr. Charles Dew, American surgeon, discovered that plasma could be separated from other components that are present in blood. In 1948, after a long time, after in 1948, the American Red Cross established a nationwide blood group program for civilians. American Red Cross is a council, is a committee, is a society, is an organization that was established in 1948, nationwide blood program for civilians. So in this way, the history of blood transfusion was established. Many scientists came into different era and contributed by their novel work to develop a successful blood bank program. Now the next one is blood group system. The blood group system in 1901, as in previous slide we see the Dr. Carl Landsteiner in 1901 who discovered the blood group system. First of all, Carl Landsteiner in 1901 ABO blood group system was given, who divided the blood group into four, A, B, AB, and O blood group based on the presence or absence of a particular antigen on the surface of red cells. If the antigen is present, on a patient's RBC, the corresponding antibody will not be present in patient's serum under normal condition. Also awarded, Carl Landsteiner got Nobel Prize in Physiology or Medicine for his work in 1930. So according to Carl Landsteiner, ABO blood groups are divided into four major groups: AB, AB, and O. This blood grouping is based on the presence or absence of particular antigens over the surface of RBC. According to Carl Landsteiner, the antigen present on RBCs, the corresponding antibody will not be present in the patient's serum. As for example, A antigen is present over the surface of RBC, so A antibody will never be produced against A antigen in the same patient, in the same person. So if the A antigen is present over the surface of RBC, so then the antibody must be B antibody, anti-B. Ruben Ottenberg, first pre-transfusion blood testing, pre mean before, transfusion mean blood transfusion testing, including blood typing and cross matching, was done by Ruben Ottenberg. And then Mount Sinai Hospital in New York. This was done in Mount Sinai Hospital in New York and also awarded by Carl Landsteiner Award in 1954. The first transfusion after doing blood typing mean blood grouping and cross matching was done by Ruben Ottenberg and also awarded by Nobel Prize in 1954. Then the blood group system. First of all, the structure of red blood cell. Biconcave, the RBCs is a biconcave hemoglobin surrounded by a cell membrane. So hemoglobin is surrounded by, as you can see in this diagram, the hemoglobin is present inside the cell and the cell membrane covered this hemoglobin. The phospholipid bilayer in which the large protein molecules are embedded. The cell membrane is a phospholipid bilayer, is made up of phospholipid bilayer in which large molecules are proteins molecules are embedded. Amphipathic amphipathic mean this phospholipid bilayer contains both of characteristic, both of qualities, both of properties of hydrophobic and hydrophilic. So this membrane is amphipathic. One surface is exposed to the surrounding plasma while the other is exposed to hemoglobin. The one surface, the outer surface is exposed to extracellular matrix, and but the inner surface is exposed to the plasma. ABH antigens here. This is an important point here. The antigens are present in two ways. Antigens may be embedded in the cell membrane or may be attached over the surface of RBCs, over the cell membrane. तो जो एंटीजेंस होते हैं वो सेल मेम्ब्रेन के ऊपर अटैच हो सकते हैं या फिर उसके अंदर एम्बेडेड फॉर्म के अंदर होंगे. हियर यू कैन सी इन दिस डायग्राम द RH एंटीजन, this the two lines represent the phospholipid bilayer and the RH antigens are embedded in the cell membrane, phospholipid bilayer. So this RH antigens are present in embedded form, but the ABH antigens like oligosaccharide chains that are attached over the cell membrane. So glycolipids mean oligosaccharide and lipids. Glycoprotein mean oligosaccharide and proteins. The protein RH antigens embedded protein embedded in the membrane with some portion exposed to the outer surface of the cell. 12 times span and display only short exterior portion of the loops. Here you can see this RH antigen is spanned over the phospholipid bilayer by 12 times, but the exterior portion, a very short exterior portion, here you can see this is this portion is displayed for the antibody as a antigens for antibody synthesis. Next one is RBC's antigens. About 96 blood group antigens have been discovered till now. 43 blood group system have been 12. ABH antigens in the AB blood group system, for example, in AB blood group system, A, B, and H antigen, 3 antigens are involved in AB blood group system. Antigens present in the environment, i.e. in animals and plants that are like blood group antigens are termed as naturally occurring antigens or environmental antigens. What is the difference between natural occurring antigens and the other antigens? The main difference, अब हमारे जो आरबीसी होते हैं उनकी सरफेस के ऊपर जिस तरह हमने कहा कि ए बी और एच एंटीजेंस मौजूद होते हैं. अब ये एंटीजेंस जो है वो सिर्फ और सिर्फ आरबीसी के ऊपर नहीं मौजूद होते. कुछ एंटीजेंस सिमिलर होते हैं जिस तरह हमने कहा कि 96 ब्लड ग्रुप एंटीजेंस हैव बीन डेवलप्ड, हैव बीन डिस्कवर्ड टिल नो. तो सो इनमें से कुछ एंटीजेंस ऐसे भी होते हैं जो अदर्स जो एनवायरमेंट्स होते हैं लाइक एनिमल्स हैं या प्लांट्स हैं उनके ऊपर भी ये मौजूद होते हैं. फॉर एग्जांपल antigen A, antigen B, and antigen P can be isolated from parasitic worms, bacteria, and pollen from flowers. So these antigens are called naturally occurring antigens or environmental antigens. The next one is blood group antibodies. Antibodies हम इनको already previous slides के अंदर discuss कर चुके हैं in detail. Here you can see 5 types of antibody in which heavy chains are different because heavy chain is involved to develop the diversity among antibodies. Immunoglobulin A carry alpha heavy chains, IgD delta heavy chains, IgE epsilon heavy chains, IgG gamma heavy chain, and IgM mug heavy chains. But the light chain in all immunoglobulins contains similar as lambda and kappa chains are similar in all 5 types of antibodies. The terminal portion of one H chain and one entire L chain heavy chains जो terminal portion है heavy chains का terminal portion of one H chain and one entire L chain function together to combine with its corresponding antigen called Fab piece. तो Fab mean fragment antigen binding. Fab mean fragment antigen binding. This one the terminal heavy or light similar in here the heavy and light. So this one is involved for antigen binding. This provides the antigen binding site. So this portion is called Fab fragment antigen binding. The FC यहाँ पे आप देख सकते हैं कि ये जो हमारी जो टर्मिनल पोर्शन है हैवी चेन्स का और लाइट चेन्स का, और इधर भी हैवी और लाइट चेन दोनों इनको हम बोलेंगे फैब फ्रैगमेंट. फैब फ्रैगमेंट मीन द फ्रैगमेंट एंटीजन बाइंडिंग. तो हेयर जो इसका जो हमारा फ्रैगमेंट क्रिस्टलाइजेबल रीजन इज कंपोज्ड ऑफ 2 heavy chains that contribute 2 or 3 C domains depending on the class of antibody. This basically depend on the type of antibody. तो FC portion जो 2 heavy chains हैं, जिस तरह यहाँ पे इसके जो एंड पे हैं, इसके जो बॉटम में जो 2 heavy chains हैं, ये मिलकर FC फ्रैगमेंट बनाते हैं. The next one is what is the difference between primary and secondary immune response. Primary immune response in which the IgM antibodies are involved, in secondary immune response IgG antibody is involved. हमने इसमें से काफी सारे ऑलरेडी हम इसको डिस्कस भी कर चुके हैं. This occur as a result of primary exposure with an antigen IgM or primary response. In secondary response this occur as a result of second and subsequent exposure of the same antigen. Responding cell is naive. Naive mean innocent और unaffected, या बिल्कुल जो न्यू सेल्स होते हैं जिनका ऑलरेडी कभी भी एक्सपोजर नहीं होता, उनको हम बोलते हैं नेव सेल. रिस्पॉन्डिंग सेल इज मेमोरी सेल. तो यहाँ पे जो सेकेंडरी इम्यून रिस्पॉन्स, इसके अंदर आपके मेमोरी सेल जो वो इन्वॉल्व होते हैं, जबकि जो इसके अंदर आपके नेव सेल, क्योंकि ये फर्स्ट प्राइमरी मीन फर्स्ट, तो इस फर्स्ट रिस्पॉन्स के अंदर आपके जो नेव सेल होते हैं वो इन्वॉल्व होते हैं. और इसका जो टाइम होता है, इसका रिस्पॉन्डिंग टाइम इज ऑफन लॉन्गर 4 to 7 days. ये 4 to 7 days के अंदर इसको रिस्पॉन्स करते हैं. सम टाइम एज लॉन्ग एज वीक और मंथ, और इसको हफ्ते और महीने भी लग सकते हैं. तो अब यहाँ पे रिस्पॉन्डिंग टाइम इज शॉर्टर 1 to 4 days ड्यू टू प्रेजेंस ऑफ मेमोरी सेल्स, क्योंकि यहाँ पे ये सेकेंडरी इम्यून रिस्पॉन्स है, यहाँ पे ऑलरेडी एक्सपोजर हो चुका होता है और उस केस के अंदर मेमोरी सेल ऑलरेडी डेवलप हो चुके होते हैं हमारी बॉडी के अंदर, और जैसे ही दोबारा एंटीजन आता है तो वही मेमोरी सेल दोबारा एक्टिवेट होकर एंटीबॉडी बनाना शुरू कर देते हैं. तो यहाँ पे इनको उस टाइम की ये विद इन 1 to 4 days के अंदर जो है जो एंटीबॉडी बनाना शुरू कर देते हैं. लेवल ऑफ एंटीबॉडी रीच पीक इन 7 to 10 days. तो but in case of secondary immune response level of antibody reach peak in 3 to 5 days. तो यहाँ पे 3 से 5 दिन लगेंगे, यहाँ से 7 से 10 दिन लगेंगे. Amount of antibodies produced depends on nature of antigen, usually produced in low amount. So actually the amount of antibody depend on the nature of antigen, but in case of primary immune response, usually low amount of antibody is produced. But in case of secondary immune response, 100 to 1000 times more antibodies are produced as compared to primary immune response. So the antibody production amount produced by standard immune response is 100 to 1000 times is greater as compared to primary immune response. Antibodies level decline rapidly. Antibody level remains high for longer period. Antibodies have lower affinity for antigen because IgM antibodies are monomer. Antibodies have greater affinity for antigens because pentamer. Here you can see, as we discussed in previous slide, that blood group A and B or AB, both three blood group carry 80% IgM antibodies and 20% IgG antibodies. But in case of blood group O, the 80% IgG antibodies and 20% IgM antibodies. So the next one is genetics of ABO. This will be discussed in second video. Thank you very much for watching this video.